cure for cancer! We've come closer to this dream by nucleic acid therapies enabling us to edit, replace, add, or inhibit genes. But is this a solution for the deadliest form of skin cancer, melanoma? Not quite. Malignant melanoma is typically associated with UV-induced DNA damage and caused by mutations in critical signaling pathway genes in pigment-producing cells. Discovery of susceptible genes and the molecular basis of pathogenesis hasn't resulted in successful nucleic acid therapy due to acquired resistance and systemic toxicity. Meanwhile, conventional therapies are stage-dependent, with tumour heterogeneity and toxic side effects posing significant barriers. What if part of the solution was discovered 100 years ago?
Welcome to the genetic basis of stuff and things.
Intratumoral immunotherapy has been regaining interest to treat malignant tumours, involving needle-based injection into multiple lesions. So what is injected? One approach is giving an enemy a makeover to fight cancer, viruses, specifically oncolytic viruses. Introducing IMLYGIC, the first and only FDA-approved viral therapy, injected directly into melanoma tumours and developed from a genetically modified herpes simplex virus type 1, HSV1, called Talimogene laherparepvec, T-VEC. Yes, the virus which causes herpes is going to save you from inoperable skin cancer. But how does the virus know a cell is cancerous versus healthy and destroy non-injected tumours too? Let's paint the scene. The protein kinase R pathway is an important pathway manipulated by cancer cells in HSV1, so replication can continue without detection by the immune system. PKR is both a scanner for cellular stress and cancer-like cellular activity, and the handbrake on cell proliferation by halting translation. Two key players in this pathway are the eukaryotic translation initiation factor 2 complex, EIF2, a protein which regulates translation, EIF2B, a protein that regulates catalytic turnover of the EIF2 complex. PKR recognizes byproducts of viral replication and cellular stress signals, autophosphorylates and activates to then phosphorylate EIF2 alpha. This increases binding affinity for EIF2B. EIF2B, therefore, cannot enable a catalytic turnover of the EIF2 complex, causing protein translation shutdown, which halts viral replication and cell proliferation. In melanoma cells, abnormal oncogenic signaling pathways block PKR activation, while inhibition of extracellular activators of PKR indirectly block the pathway. This means stress induced by aberrant proliferation cannot be detected, preventing shutdown of translation to enable the melanoma cells to keep replicating. Like melanoma cells, HSV1 has devised strategies to also avoid detection by PKR and the immune system. So how has HSV1 turned into T-VEC to take down melanoma? 4 key steps occurred in the makeover. The HSV1 neurovirulence protein, infected cell protein 34.5, encoded by the gamma 34.5 gene, activates PP1 alpha phosphatase, which dephosphorylates EIF2 alpha, so EIF2B is no longer able to bind to it, preventing shutdown of protein translation. Mutants with both copies of the gamma 34.5 gene deleted fail to replicate in normal cells because the active PKR pathway causes cessation of protein synthesis prior to completion of the viral life cycle. The ICP34.5 mutants can replicate within cancer cells because in these cells the PKR pathway is suppressed to enable unregulated growth. Next, 2 copies of the human granulocyte macrophage colony stimulating factor gene, GM-CSF, were inserted for local expression to occur in the tumour microenvironment. This promotes dendritic cell recruitment and accumulation, antigen-presenting cell function and activation, and primes and promotes cytotoxic T-cell responses to tumour-associated antigens. Next in the makeover process, a spontaneous mutation occurred. Deletion of the infected cell protein 47 gene, followed by translocation of the US11 gene to be regulated by the alpha-47 promoter. The ICP47 protein binds to a transporter associated with antigen processing to down-regulate expression of viral antigens on the surface of infected cells, preventing CD8 plus T-cell immune responses. Deletion of the ICP47 gene causes tumor antigens from within the cancer cells infected with the virus to be presented on the cell surface. This generates specific T-cell responses to antigens present in each individual tumour cell, overcoming tumour heterogeneity and unmasking hidden melanoma cells. Lastly, the US11 gene encodes an RNA-binding protein which binds to PKR, so it can't phosphorylate EIF2 alpha, thus preventing the shutdown of translation. US11 is normally controlled by a stringent late promoter, so the protein is not made in healthy cells infected with ICP34.5 mutants. However, the translocation mutation resulting in regulation by the alpha-47 promoter causes expression of US11 as an immediate early gene. The US11 protein is thus able to block the PKR pathway before shutdown of translation. This enables enhanced replication of ICP34.5 deleted viruses in tumour cells without compromising reduced pathogenicity. T-VEC lysing melanoma cells then promotes anti-tumour immune responses by causing local release of cytokines, IFNs, danger-associated molecular pattern factors, pathogen-associated molecular pattern factors, and toll-like receptor agonists. Tumour antigens previously not presented on the cell surface are also released, which can prime de novo anti-tumour CD8 plus T-cell responses. Meanwhile, a systemic anti-tumour effect and complete immune response occurs via activation of DAMP pathways. Intra- and intertumoral heterogeneity is thus addressed by primed T-cells inducing a systemic polyclonal anti-tumour response. Reduced total tumour burden in injected and uninjected lesions in the T-VEC Stage 3 clinical trial was evidence of both regional and systemic immune responses. The success of IMLYGIC in patients with unresectable malignant melanoma paints a promising picture for development of future oncolytic therapies to target other cancers. Who knew genetic manipulation could turn viruses into a weapon against cancer?